You're listening to The Interactome, a podcast by a group of young researchers who want to connect you to the world of science by sharing their stories and perspectives. Just in case their bosses are listening, they want to remind you that the opinions expressed here are their own. They also want to remind you not to take anything they say as medical or professional advice, as they are not doctors. Not yet, anyway. Stay tuned about that. And, without further ado, welcome to the Interactome.
Hi everyone, and welcome back to the Interactome. We are back with another, I would say, internal team episode. We don't have a guest this week, but another really interesting topic nevertheless. So I'm Natalie. I am a science comms expert if you're new to the show, and I'll pass it over to my co-hosts to do intros, and then we'll kick it off.
Hey everyone, I'm Joe, currently doing my MD and PhD, doing some fun metabolism research, but today we're going to get to learn some interesting stuff related to, more honestly, in my opinion, to the MD side of things, but I'll let Sam give his input on that.
Hi, I'm Sam. I am a currently unemployed process engineer, which is, I've been vague about what I do in the past. I still will not be talking about any former employers. For the record, they're all lovely. It's a long story. But the industry's in a weird place right now. But because there's no one in particular to get mad at me for talking about this stuff, I don't have to clear it with anyone. I'm going to talk vaguely about what that means, what I've been doing.
Heck yeah.
Well, actually, I'm going to talk very clearly about the not proprietary stuff. You're not going to hear about any projects I worked on, but you're going to hear about the kinds of things I've been doing. And I guess, kind of more simply, what is process engineering? What is, well, process science? I guess I'm technically a scientist. It's complicated. It's very much, and it's for a reason, and that's because that's the in-between of, essentially once the people in research who are very clearly scientists figure out how to make something, it's the process development, which is like process engineering, process sciences department job to figure out how to make medications.
And I imagine this means like making it in an optimized way, right? Like as your process engineering, making it so like you are saving the most money, making sure things are like made the fastest, highest quality. But I'm sure
it's even sometimes more fundamental than that, particularly for the class of molecules I'm familiar with, which are biotherapeutics. I don't know about small molecules, so like how that starts. I know about them. I've taken plenty of biochemistry courses. I've worked on.
He's like, I've never heard of a small molecule in my life.
How before we get into it, how would you differentiate between a small molecule versus a biotherapeutic?
So a small molecule,
for listeners.
So there's like a size cutoff that people use. I think it's something like 1,000 times the mass of a hydrogen atom or something. But for listeners, the big, and also realistically, the big thing is like a small molecule tends to be typically like a chemical that is the sort of thing that's synthesized by organic chemists. It is, this is kind of off color, but like I couldn't do Breaking Bad. That's those sorts of molecules, those are small molecules, right? Amphetamines, which are legitimately also used as therapeutics, are small molecules, as is something like Tylenol, as is something like a lot of like, especially like early chemotherapeutics, allergy medication, SSRIs, statins. Those are all small molecules. Most of the drugs that you're familiar with, most medications you're familiar with, you have in your medicine cabinet that you can swallow as a pill are small molecules. That's actually, that swallow as a pill bit is often very important. A small molecule is more easily absorbed into the bloodstream through your digestive system, through your intestines, that sort of thing, because they're small enough to permeate the barriers there. A biotherapeutic, on the other hand, is something like insulin, right, that would need to be injected. So the first biotherapeutic was insulin. But the and like there's other things that probably would fall under that category if you go back in history. Based on the molecules involved, a lot of early vaccines are also technically biotherapeutics. They're biomolecules. They tend to be synthesized by a living organism, whether that's something like a microorganism like bacteria or yeast or cells of a mammalian organism, which are typically used to make biotherapeutics. There's also, especially in the past, there were biotherapeutics that were directly sourced from animal products. So insulin, before it was produced, quote unquote, like what we call recombinantly, or like in bacteria or in an organism that didn't naturally produce like human insulin, it was often harvested from animals. So early biotherapeutics were, tended to be harvested from animals, actually, which is exactly as nasty as it sounds. And also,
would they undergo like any like changes, or was it just like a really raw kind of like, okay, animal to whatever the I'm trying to think of like what year.
Well, I mean,
guess
if
it doesn't
you're thinking
matter.
like
This isn't a
really
history
way
podcast,
back
but I'm
into
curious.
history, I mean, like, no, I mean,
Yeah.
this would be like, I want to say like the earliest use of like, I think porcine insulin, so insulin from pigs, was like early to mid 20th century. Like, we didn't have the ability to do any sort of manipulations with these things. And that came with consequences. I don't know if necessarily with insulin, but we're going to get into like a lot of my job has been making things that are high purity, that are free of contaminants, that are relatively uniform, which is hard to do with a biotherapeutic, and even harder if you're coming from a living organism source, like a mammalian source like that. But I think if anything, you could tell from that, even that question alone, they're kind of a pain in the butt. I'm gonna be honest, like biotherapeutics versus something where, you've got some guy from DuPont who's got a giant reactor and the whole thing is, a hermetically sealed thing, versus something that's coming from organisms that need to eat and breathe, even if they're single-celled organisms. You end up, it's a very different beast. It's challenging, and I love that challenge. So I guess the main question there is, why bother, right? And a lot of that is because if you're getting a larger molecule that is of a biological origin, you can end up with things that are more specific, that can target specific parts of the body or make changes that a small molecule never could. This includes things like gene therapies, where you're editing someone's genome. Most commonly, especially today in the pharmaceutical industry, especially in terms of like market share, antibodies are huge. And the neat thing about antibodies is organisms, humans included, our immune systems are really, really good at targeting specific stuff. And that specificity, especially when combined with engineering, done by, skilled folks in the research teams, you can end up with an antibody that targets very, very specific cells or, other phenomena in the body for degradation by the innate immune system, not innate, but our own immune systems. And so that's like one of the benefits of it, is you can go, hey, there's this very, very specific problem. We're going to engineer a very, very specific solution to it, versus a small molecule, which might not be big enough to be super specific. You can end up with, especially early small molecules, like some of those, I want to say aspirin is like actually has like really wide ranging effects on your body, because it has a lot of things it'll stick to. It's not specific. It's not targeted. It's cheap. It's easy to manufacture, and it's effective. But that also comes with side effects, relatively minor ones if you're taking a reasonable dose. But you can create a much better, more specific class of molecule that can do things like, say target like arthritis, for example. You can target inflammation, cancer. You can also upregulate things. There's classes of molecules that can cause you to produce more or less of a growth factor, and if you have a deficiency or there's a problem with your body where you're not making enough of something, that can be remedied, which is really, really hard to do with a small molecule, because you have to find essentially a 3-toothed key that can fit one lock out of millions. Like it's like you have to really, versus like you're saying, hey, I want to build a box that only, like, if you shrink wrap something, you vacuum form something, you take that off. It's a very specific lock and key that fits a very large thing. And because we can hijack biology to make stuff that's that specific, we can create these really cool systems.
I have a cool biotherapeutic I wanted to talk about that is kind of becoming hot now. So there's this is actually something that we're researching right now. Originally, this was developed in an attempt to help treat muscular dystrophy, but it's now being used to try and help people who are like losing muscle with age. But essentially, have you guys ever seen the memes of the really beefy cows?
Yeah.
So okay, so there are memes of really buff cows on like Instagram and stuff like that, and
Wait, that's hilarious.
Yeah. So
this is a real thing.
Yeah, so they have a mutation in a gene called myostatin. Myostatin is a hormone that tells the body like when to stop making muscles grow, essentially. So like it negatively controls muscle size. And so these guys don't have myostatin that works, so they get really freaking beefy. And now and so the thing that actually historically there have been myostatin inhibitors that like bind to myostatin, which like what Sam has been talking about does it's not a small molecule, it's a hormone, but it does a lot of different things in the body. And so people who were using myostatin inhibitors for bodybuilding and stuff like that were getting a lot of issues like for example, like heart issues, reproductive issues, like all those sorts of things. But more recently, a monoclonal antibody has been developed initially for treating muscular dystrophy, a degenerative muscle disease where people like young kids lose muscle and muscle function. But it wasn't shown to be very effective there, but it's now being explored to basically dope older people when they lose muscle due to sarcopenia or like being stuck in a hospital for a really long time or things like that. And so you have this monoclonal antibody that is very specific to the receptor for myostatin that's important for shutting down muscle growth. So if you shut that down, you theoretically turn on muscle like growth. And so that's being evaluated now. And so the thought is, and we'll see whether, like, it's in clinical trials right now. Actually, I think some interesting clinical trial data is coming out this summer. But we'll see whether it helps patients or not in this context. Obviously, that's why we do studies to see whether it works, and it will need to get approved for all these kinds of things. We'll get into that later. But that was just one that I thought like blew me out of the water when I first learned about it. And I get to work with it now. It's really interesting. So yeah,
that's very cool, Joe.
And Sam's responsible for making things like that. So
yeah,
thank you, Sam.
I again cannot really elaborate on what I have or have not worked with. Joe does have an uncanny ability to make me kind of sit there and nod and be like, but the so if you try to make something like that and you hire somebody like that, like me, to try to make something like that and not talk too much about it on a podcast specifically, there's kind of there's a process that happens, right? So one of the things that I think is important to note with the start here is that a research production line doesn't always get the highest purity product. And by highest purity, I mean something you'd be comfortable injecting into a human being. And there's a lot of reasons for this. For starters...
Can I just backtrack and ask what a research process line is?
I'm being vague for a reason because companies do this differently. So,
okay.
What happens prior to something hitting the process development department can be anything. It could be anything. It could be there's a dedicated team that is like, hey, we want to make XYZ thing. Here's the DNA sequence. Go do that for us. That tends to be more like a PD department does. It tends to be folks who are working like what's called like a core lab, and folks will be producing that in relatively large scales, but also like not typically I would say you don't waste time on things like purity. So there's kind of a cost-benefit analysis where if it's close enough, it's close enough. You throw it in some mice, you see it if it works. If something happens to the mice, then you assess maybe if there's you're not going to be like filthy with it, but
you're not worried about making something that's like pharmaceutical grade at that point,
or even like injected into like a monkey grade. Like a lot of times that's like a discussion versus like the like full. Okay, before you inject into mice, that already has to be thought about. There's the first baseline thing is you're doing in vitro experiments. You're doing stuff in a test tube. That good enough's good enough. It's not always. Speaking from context of doing, my particular realm of expertise is protein purification. So proteins being the main component of a lot of biotherapeutics. Antibodies are proteins. The outsides of viruses are often protein. They can sometimes have a coating of fats. There are folks who do like nucleic acid stuff, which is fascinating to me, and I'd love to try it in the future. But purification.
You're a protein guy.
I'm a protein guy, and I'm particularly figure out purifying things, isolating stuff, and
proteins are your proteins.
And like sometimes there's trouble with that, and even in an in vitro context, you don't have it high enough purity, but or you don't always have the same stuff. So proteins get modified; they also break down. So if you say leave it at room temperature for long enough, it's not stable. It's going to break into pieces, and then those pieces are not going to be indicative of what you're working with. That's typically for like research molecules. Usually, anything that goes into a person is robust enough that you can kind of have some temperature range there, where at least for research is good enough. This is all to say there's kind of grades of things here, and the end goal that we're going towards is it's as darn close to one molecule. It's got very little other stuff in it that doesn't need to be there. You know what's in it well enough to tell the FDA this is what is in it, and you send the FDA a giant packet of paper saying this is what we know to be in there, this is what we think is going on here, this is what we did and why, and then also it's pure enough to be injected into a human being, which there is a lot that, and we'll get there. But if we're going to back up, it's the protein. Good enough. Some people will be like, hey, I have what's called like an affinity tag. So like, it's just like, you put a thing on there, it's like, yeah, it's essentially, one of them gets Marco, the other one gets Polo. It's like, hey, Marco, Polo, okay, high fives, they're stuck together now. We can pull it out of solution. It's like you just stick Marco onto the protein.
That's a great example.
Yeah, you play Marco Polo with proteins, except they once you get together, once they find each other, they're stuck.
I think another good analogy is like a magnet.
Like a magnet. Yeah. But I think a magnet, though, is a funny one, Joe, because a magnet, if you have like metal shavings, you have a screw. Like if you ever like take a magnet and you try to stick another magnet to it, you're gonna get the other magnet. You're also gonna get like anything else metallic.
That's a good point. That's a good point.
Whereas an affinity tag is something that's intended to not stick to the other stuff.
Yeah.
It doesn't always work that way, but that's your goal. Affinity being like, if you have affinity to something, you like it, you're really into it. They really like each other, they stick together, and they don't tend to come apart unless you either find something it likes even more, or you break part of that, like you denature it, falls apart, and it releases it. Anyway, sometimes you just do that, and it's done. That's your research process. And there was this guy I heard talking once who's like, yeah, the perfect process is 2 steps. One to like capture your product, and one to catch your mistakes. But you still need to catch your mistakes for a pharmaceutical grade one versus research, you don't. So, well, sometimes it's good enough. Good enough is good enough, and you just check it and you like kind of ballpark, you'll run a gel or something. Like, yeah, that looks like the right size, use it. Obviously that doesn't fly for a pharmaceutical, for a lot of reasons. So, that's why we have to do this. Research, though, tends to work with a molecule. Also, the other thing is some classes of molecules, you can have, like, lots of contaminants in there. You can sometimes just work with, like, a slurry of stuff. I've seen, and like if it's concentrated enough, you don't really even purify it much. There's some zany things that happen in research, and then it tends to get to like an early stage, like preclinical was what we call it, where you do like preclinical research. So you're injecting to mice, you're injecting to monkeys, and so on. And often that's when a process development team first gets something, when the experiment has become expensive enough and involves ethical considerations enough that you can't just be messing around.
And I have a clarification question. In general, though, what you're saying about research grade things versus like pharmaceutical grade things, is if let's say I want to make myself really buff by getting this monoclonal antibody, I should not be buying it from a store that sells research grade.
No,
I should be getting it prescribed to me, and then getting the pharmaceutical grade if it's indicated for me.
Yes, generally. In general, yes. I was going to say, though, generally, research grade can mean a lot of different things. So if you're in a lab working with a research grade product, there's a chance that it's made exactly the same way, but the paperwork's not as thick. So, there's some processes, particularly for relatively simple stuff, like buffers or so on, where, like, the research grade product is made in the exact same lab as the pharmaceutical grade, like, manufacturing precursors, for example. Like, small, like, industrial molecules. I've worked with things where like there's the pharmaceutical grade and the research grade, and it just costs more money because there's a longer paper trail. That sounds dismissive, but like I've seen things happen when you don't have that paper trail, when you don't know who made it, why, what the contaminants might be. Specifications are really, really important, and you can get lot-to-lot variability with research-grade materials that can be pretty egregious, depending on how you're checking. Again, pharmaceutical process, I would say, is one of the, like, most stringent things we have in terms of, like, manufacturing processes possibly on the planet. It's, like, up there with, like, aerospace in terms of, like, how long it takes to make something, how many regulators are involved, how many different people have their eyes on something, how many opportunities it has for somebody to say, hey, this is not safe, not effective, too expensive for... And, like, okay, you laugh at too expensive, there's a lot of different things there, but, like, if we're spending... You could get really silly with it and say, hey, you could have a drug that treats 5 people and you're spending, like, half of a small country's GDP on it, and, like, that's not really the best decision to be making as a society, I don't think. From a utilitarian perspective, that can get really thorny, but there's a lot of drugs that are made for like rare indications where it's like, is anyone ever going to foot the bill for this, and does anyone want to? And it doesn't mean that the drug that disease won't get treated. It just means that, hey, maybe this approach is completely wrong.
Or it's not feasible for the company to make it.
It's not feasible for the company to make it, not physically, not economically. There's a lot of like challenges that come up. It's not a simple equation. And a lot of that starts to happen once a process team gets it. There's a huge handoff that happens between research. Like there's like the RTD transition, the research to development transition, which is huge. That's like a major corporate decision. It takes like months to work through. There's huge data packets that lots of different people are looking through and saying, hey, is this drug something that we want to start testing in humans? Is it something we want to start testing in monkeys actually first, usually? Or that I say monkeys because that tends to be the sorts of things I've seen, rhesus monkeys, because they're so close to humans. On the other hand, they're expensive. There's a lot of ethical considerations. Nobody wants to, go out there testing things on monkeys that is not going to help a person. Which is something to be said about animal testing, is it's really expensive. It's hard on researchers. It's generally a difficult, time-consuming thing. It tends to only happen when we think it's going to actually save lives. And at least in the pharmaceutical industry. I don't know, maybe there's other things happening elsewhere in the world. But so there's a huge decision that happens there. Like, and that's all before someone like me would even see it. Typically we're aware of things, like we'll chat at lunch, like, hey, how's this project going on in research? Oh, that sounds promising. I wonder how that's going to work. We're like, well, okay, what are the challenges going to be? I might spend a couple hours looking through papers being like, hey, what might we need to anticipate if this is going to happen? And then then we actually get to the process of actually making something. There's the
Yeah.
are we going to make it? Which I think is probably reassuring people to know that if they're taking a drug, there's a lot of thought that goes into it. But yeah, so you've got your hypothetical idea, you've got your hypothetical drug. Now, how do we make it an actual drug and not just something that, someone managed to figure out how to make in a day to test in a test tube?
Yeah.
And so there's two parts of pretty much every process. They call it like upstream and downstream, right? Upstream comes first,
Okay.
downstream comes afterwards. I do what's called downstream. I've always been like a downstream processing person. I do protein purification, filtration. There's clarification and harvesting. And what are we clarifying? What are we harvesting? What's upstream? Where's this start? And that's cells, usually. So some therapeutics, like nucleic acid therapeutics, can be made in vitro. There's like in vitro transcription, in vitro translation, which is essentially taking the parts of a cell that do that, putting them in a tube, and then having them do those things. You put in reactions, you make a reactor, which is simpler if you can get it to work. So like a lot of like RNA stuff I think is done that way, if I understand properly. But proteins, for now, we don't have in vitro protein machinery. You need a living thing to make it. This is the first probably big decision that happens in any process thing, is what are we going to use to make the thing? For a lot of biotherapeutics that are relatively complicated, you need not only the protein, but also post-translational modifications, changes that are made to the surface of that protein that allow it to interact with things differently. And the different cell lines do that differently, and the typical ones you get are human cells. So there are certain human cell lines that are immortal. They grow in vats, and they are usually based off of like cancer cell lines or stem cell lines, and those tend to be like kind of the gold standard for something going to humans because it's human cell to human cell. Antibodies are often produced in CHO cells, which is short for Chinese hamster ovary cells. So I call it a big old vat of hamster cancer, which it rolls off the tongue. It really sounds nasty. To be fair,
Say that 5 times fast.
yeah.
Big ol' vat of hamster cancer. Big ol' vat of...
These all sound nasty. They kind of
Big ol' vat of hamster cancer.
they don't smell great. I'm gonna be honest, but
what are you just like going in and like?
no. Well,
wafting
once
with
it gets to me, you have to crack the thing open a lot of times. Well, okay,
That's true.
Okay, that's not true.
true.
That's when I was working on research stuff. Everything
Okay.
tends to be closed systems, and but it's not sterile at that point, so it could be open.
Yeah.
So it's not sterile because it's cells. There's living stuff in there. There could be viruses. There are other different lines. There's also plant cell lines and there's insect cell lines. Plants tend to be relatively rare, but actually people have started using whole plants, tobacco plants in particular.
Fascinating.
Yeah, I've heard a lot of stuff about this. You tend to throw leaves in a blender as your first step. It's like wild.
Interesting.
Yeah, which is cool. I haven't worked on that yet. I'd love to try. It sounds really interesting. And then the other one being insect cell lines. I don't remember
I've worked with some insect cell lines before.
Yeah.
SF9 is a good
SF9,
one.
yeah. I don't
good
remember
one.
what the organism is, though, what the original.
I think it was some kind of beetle, if I remember correctly.
Maybe.
But either way, they're cool. They like just they float around. They don't stick to a dish. From what I remember.
So that's suspension versus adherent cells.
Oh boy, so many cells. What the cell?
So now you've got the type of cell, and then you ask, is it floating in a tube or is it stuck to something? So things that like in tissue-like formations like are stuck to the sides of things is more like natural for a multicellular organism. You can also make things out of yeast and bacteria, by the way. So that's yeast, like baker's yeast, pretty much, or usually E. coli is used for expression. And then you have adherent versus suspension. Adherent's a pain in the butt because you can't just make the tube bigger. You have to scale up whatever it's sticking to. And these are the sorts of considerations that come into process design, because if you have to scale up something that's like got like a sheet of gunk stuck to it, it's a pain in the butt. You realistically just want a big old vat of something with an impeller in the middle of it, and you put in nutrients and gas, and
it goes.
That's your game. You just start small and you get big, and that's how you start. It's easy, relatively. You put it in a sealed container. There's these big metal tubs, essentially with a big propeller in the bottom of it, and that stirs it up, and it's just growing like beer or kombucha or something like that. You know? Like, really, I mean, it's the same idea. It's it's practically, and the technology looks very similar. Like, if you've seen, there's a few labs around in Boston that do scale, like process scalp. You can actually walk by them. I think Amgen's got one, and it looks just like a brewery, a really clean brewery, is what it looks like. But they're growing up probably that hamster cancer I'm talking about. Each one of those also has different nutrients you have to feed it, and so on. And generally speaking, the whole goal with any sort of process, any sort of engineering, is keep it simple, silly, right? You want the simplest system possible. If you can make it in E. coli, make it in E. coli. If you can make it in yeast, make it in yeast. If you can make it in suspension, you don't want to bother with adherent. You want something to be as simple as possible. It's like cooking. Why would you bother, like carefully sauteing something, 2 minutes on one side, 2 minutes on the other, take it out, put it in the oven, wait 20 minutes, take it out, pour something else in, put it back in. Let's pain in the butt. If you could just like throw it in the microwave and hit the add 30 seconds button, that's what you want. Same thing goes for manufacturing. You want the microwave dinner version of whatever you're doing. Speaking of microwave dinners, that nice plastic seal on top of that, you really just want everything to be sealed. You want everything to be enclosed. You don't want stuff to be able to get into it, because that can contaminate it. When you're talking with Upstream, from my understanding, is contamination tends to just kill the cells. It's less of a concern for the end product, because it's my job to make sure that any of those contaminants are out of there. But your cells die, cells, just starting that cell line can cost an ungodly amount of money. It's like if that thing dies, it's like flushing a Lamborghini down the toilet. It's not even like that's the kind of order of magnitude you're talking about.
That's what we should have for the episode cover. A picture of Sam flushing a Lamborghini down the toilet.
Don't want
I agree with that.
So once you've done this very expensive, this takes like months sometimes to scale up. So what you start with is little like flask, like the little, the little conical little cone-shaped flasks that you see in like your Halloween aisle at Michael's or whatever, or like where it's like here's your science tube. Yeah, you start with one of those, and then you scale up to the giant room-sized beer reactor vat looking things. Then you have that, and now you have a vat of hamster cancer that also happens to contain a heck of a lot of your drug, right? Yeah. And now you have to do several things. So in the case of CHO cells, that hamster cancer I've been joking about, a lot of those things are secreted, so you avoid one of the worst problems, which is get rid of all the other crap in there, right? You don't want to have to get rid of it. So a lot of drugs, you have to lyse cells, you have to split them open. There's a lot of different ways to do that. Sometimes it involves a detergent, you got to get rid of that. But the other thing is, now you end up with just cell goo. So for antibodies and that hamster cancer stuff, you just run that through a screen and it just traps all the cells. You don't bother to split them open, and you end up with a bottle of what looks like orange Gatorade and smells like vomit. Again, I'm upselling Upstream work is really difficult, and I am not particularly good at it. So I'm making all these jokes about it, but that's incredibly, incredibly valuable stuff that can save your life.
Just to reiterate, at this point in process development, you have hamster cancer Gatorade. That's the cost of a Lamborghini.
Oh, way more than a Lamborghini, especially if you're going to start selling that stuff. Well, it takes time and effort and skilled people. And remember how I said like research grade versus pharmaceutical grade? Pharmaceutical grade components to feed those hamster cells. You have time in a clean room, potentially. Usually, I mean, you have time in a clean room. That's a huge facility. It's custom built for all of this. You're either renting time from another company to do that, or you've built this facility yourself. As a company, that's incredibly expensive. Yeah. And we're talking, like parts per million in terms of dust. You have people walking around in what are called bunny suits, like, these like, the joking, the Tyvek suits that don't allow any like hair or things to come off of you. and potentially contaminate it. You've stressed over taking care of these cells, which is like, taking care of the fish tank from hell. And you've now accomplished this, and you now have this vat of stuff. Now, the thing is, you don't want to inject the nasty Gatorade into somebody. That's not the drug. There's a lot of reasons why it's not the drug. For starters, it can contain anything else that the cells secreted. It also contains all the nutrients it could have been consuming. It could contain bacterial or viral contaminants that somehow got in there. You want to get rid of all of those things. And some of those things are big, like viruses and bacteria, and can be removed by filters. You literally just filter it now more finely with something else. It's the same thing as... It's not chemical. Depending on the filtration system, it could be relatively similar to a fish tank filter. It could be relatively similar to, those kinds of filters that you'd have in like a water bottle when you like have like one of those camping water bottles. But that tends to be unusual. Or you could have like a fine like like a fine surface or
mesh or something.
It's like a mesh, but the holes are nanometers across.
Yeah.
At the biggest, right? Because the biggest thing you let through, that's the size of the screen. And so you have to do that. So typically your first step, if you have a lot of gunk in there, is to run it through like a dense, it's called a depth filter. It looks like the stuffing of a plushie. It's this like, it's highly engineered, super sterile, highly controlled manufacturing, sometimes different chemically. It's probably not that kind of polymer, but like depth filtration is used in like polishing filters and fish tanks and stuff. Like I've got media for that for my fish tank in the other room, but it's not exactly the same. But also like anyway, so you can start with a filter. If it's relatively pure, you can start with that affinity thing, right? So the neat thing with antibodies in particular is antibodies are made to stick to something in particular, right? So if I have an antibody against, a cancer cell, you can take that little receptor and stick it to a column and it'll catch onto that. Or, here's the thing about antibodies. So one end sticks to the thing you're trying to detect, the other end sticks to immune cells. And so you could take the end that is meant to... You can take the part of the immune cell that sticks to the antibody. I believe that's what it is. I think it's what protein A, protein G are. And they stick to the common part of an antibody that the immune system is supposed to recognize. Now, of those two, you can imagine the special one for your very specific thing is way more expensive than what sticks to every single antibody. So everyone just uses that. And so that's your first step. Typically, your first step is try to exclude everything else humanly possible. This does a lot of different things, but for an affinity step like that, you also concentrate it. You have just what you want. Then typically you'll do some kind of polishing step, they call it. So you can separate things based on size. So if you have something that's too big or too small but happen to stick to the antibody thing, so say it's a degraded antibody, it's too small. Say it's like a bunch of them glommed together, it's too big. Size would work great. Sometimes charge is used to separate things too. That works really well if you have something that might be binding to it, like DNA, to separate that out as well. Yeah, and so there's a challenge. It's kind of like saying, how can I get this and not that? And if you've ever had to separate out any sorts of things before, I mean, I have a giant Lego collection, so size is really useful for that, right? I mean, if you want to start sorting things.
It's true. I've been to his apartment, and it is an
behind
impressive
Agreed.
Lego collection.
It's a lot of stuff. But yeah, if you have to start sorting something, you kind of have to start with like, how do I start? What do I want to separate things by? And that's how you start to do purification. So that's the technical side of things. And all of that now has to be done in a sterile environment with as enclosed a system as possible. You have to then also filter for things like viruses and bacteria. Again, like I said, you separate by size, but you do that like multiple times just in case something got in. Usually like kind of towards the beginning and then at the end, definitely. So like sterile filtration, we'll call it. Now, if you're working with something really big, like a virus that you'd use for like a gene therapy, or there's people who are trying to make like more exotic stuff using what are they? Like vesicles, they call them, which are just big fat bubbles and stuff like that. Those are big. The bigger something is, the harder it is to remove viruses and bacteria, because those tend to be big. On the other hand, viruses and bacteria don't love soap. This is actually a way that you can get rid of things in a manufacturing process. You add in detergent. Like when you wash your hands with soap and water, that soap is doing a lot. And this is like an aside that's like, hey, life advice: soap does a ton. We use detergents, like manufacturing grade detergents, to remove bacteria and viruses from a pharmaceutical that's going to be injected in somebody, and it gets like 1,000 times reduction in those sorts of things. And this is also true if you read public health papers. It can get rid of some real nasties, to the point where, like, if I have something nasty in my house, or like I'm trying to clean it up, like the first thing I go to isn't bleach, it's dish soap. Like, it'll get rid of a lot of stuff. So, like, fun fact, soap is really strong, really powerful.
Sam?
Yeah?
I really appreciate you getting up on your soapbox.
No notes.
I mean,
Stunning.
Yeah.
Process engineering
the
soapbox.
Detergents are
Name
great.
of the
Detergents
episode.
are phenomenal. The only problem with detergents is something you inject into somebody is you have to get rid of them. You don't want to inject detergents into your body. Do not do that. It's actually relatively easy to do. It's small.
But Tide Pods, they're so delicious.
No, Natalie.
This
again.
We can edit that out. Sorry, Pickell.
We will need a number of therapeutics to help Natalie if she ingests a Tide Pod.
Anyway, I don't know how to solve that problem. I'm not a doctor. Joe, you're going to med school. Maybe you could talk about how to solve that issue.
I'm not a doctor yet, so don't come to me for medical advice.
Joe, do I eat the
No.
Tide Pod or no? I have them in my apartment.
Depends
I'm really
on what you
thinking
care about. Quality versus quantity of life.
I don't think that's tasty, Natalie. So the Tide Pods aside. So I talk about detergents going into drugs. We need to get rid of that. There's a lot of things that we need to get rid of. We need to get rid of bacteria. We need to get rid of other things. How do you make sure something is good enough to be injected into a person? So all of this process aside, you can have the best process in the world. The FDA will not let you put that into a person until you can prove that it has... First off, you test the process. You will do things where you spike in a virus that stands in for something potentially dangerous, and you just test if it gets rid of it. You also...
I didn't know that
did
either.
not know
That's
that.
very
That's
cool.
very-
It's very standard protocol. So you say, hey, this thing, this filter we're using, that's how I can say, oh, you can get rid of... That the detergent gets rid of X fold of the thing. Is that gets tested. Detergent gets tested. The purification steps get tested. The filtration gets tested. And you spike in a bacteria, you spike in a virus, you spike in whatever it is you're afraid it could be there, and then you watch it go away. So that's thing number one.
That's really cool.
Yeah. So and
that's good science too.
Yeah, and you can get rid of like millions of times. So everything's like fold reduction. So as scary as it sounds, fun fact: if you get one of a virus in you, it's probably not going to do anything. Your immune system's probably going to get rid of it before it multiplies too much. This is like if you have ever been in a subway tunnel, like you have inhaled things that you would not like. People have tested the air in there, and there are things in there that you would be horrified by, and nothing happens because it's not a lot. Dose makes the poison. The number of viral particles you consume dictates how dangerous, how sick you could get. So your goal is to like reduce things by like 1 million or so times in manufacturing processes, because it's in a clean room. It shouldn't be in the process to begin with. Cool. You do that, then you test it. So you say it works, whatever, then you test the drug itself. You pull vials that you pop them open, can't be used again now, they can't be used, and they get tested. So you test for any contaminant you think could possibly be there and potentially be dangerous, and this depends on the drug. So you'll obviously test for things like bacteria. You'll test
cell goop.
for cell goop. You'll test for just junk. like optical density. People actually visually inspect every vial in a lot of these lines. So like there are people who are just trained for having good eyes, and they hold it up next to a completely blank sheet of paper and they say, is there anything else in there? Is it the right color? But one of them that actually is like, so one example of a test, because you can't run through all of them. These test panels, this episode's already getting long. You would be here all day if I ran through everything that could possibly be on a testing panel. And it is based off of what we think the risk could be. Obviously there's one very common one, and that is something called endotoxin, which is how you test for bacteria. Endotoxin being toxins that are inside something, they're inside of bacterial cells. They tend to be fever-causing. They're called pyrogens sometimes because they cause fever. Pyro, like, a pyromaniac, someone who sets things on fire. Pyrogen is something that makes things hot. It gives you a fever. That's what it is. It's from back before we really understood what they were. That's where the name comes from. And that is done by endotoxin testing. And the way this works is you take an extract from horseshoe crab blood. And horseshoe crabs' immune systems are adept at finding pathogens, and when they do it, it changes color. So horseshoe crab blood is blue, but the stuff is yellow when you get it, and the more yellow it gets, the more likely it is that there's something like in it. And so you take these sealed kits that are made by companies that extract them from
the crabs.
From the crabs. There's a Latin name. It's LAL is what the tests are called. It's like limulus immunocyte lysate or something. It doesn't. But one of those words is horseshoe crab. Yeah. And you take that thing, you slot into a machine that's specially designed to just read the color of this thing. You put the stuff in there, you wait 15 minutes, and if it turns yellow, you're screwed. So that means like something has gone wrong. But it's a huge industry and it's super important. They used to do this by injecting rabbits. They finally moved to the horseshoe crab blood. It used to be, hey, did the rabbit die? That was the test. And we did this because there was drug contamination and it would give people fever. A lot of times children, people were more susceptible to this. And so the horseshoe crabs, by the way, usually survive the process of, it's like they bleed them for this and then they put them back in the wild. It's relatively contentious. There may be, I believe there are synthetic alternatives, but when it comes to are you going to give someone a toxin and a medication, people are really afraid to do that for obvious reasons, to really mess with that. Like, you don't want that to happen. And so it's been kind of iffy on moving to a non-horseshoe crab alternative. But regardless, that is one test out of an ungodly many. So, yeah, and again, it depends on your product, too. So, like, there's all kinds of stuff. There's, like, efficacy testing. You have, like, cells, and you test if it does the thing to the cell. There's size testing. There's, like, you're just checking the size of the molecule. There's many ways to do that. I mean, they get up there about everything. Like, how much is in the bottle. Like, anything that could potentially indicate a problem is tested. And let me tell you, when you're waiting for a clinical trial to clear testing, it's nerve-wracking. Like, you throw a party when that thing clears. Because there are so many things that, no matter how careful you are, you still need the test to pass. It's rare that something goes wrong, but you still need to make sure. And it's essential that everything in it goes off without a hitch. And this can be months of planning, more dollars than I've ever seen in my bank account by orders of magnitude. And yeah, it's a lot of everything. And then you can run a clinical trial with that material. So the first thing you want to do once you've accomplished all of that is you test in a small group of people to either make sure it's safe, or if it's a drug that is going to is not necessarily something you want to get if you're not sick. Like a chemotherapeutic, like gene therapies, which will edit your genome. You'll put it into a small population of patients. You'll see if it works, as well as whether or not it's safe. Yeah, and so I think Joe, you might have some feedback about that one, because I don't do that. I just get to see the results, and whether or not all of that effort and nerve-wracking waiting was even worth it.
Yeah, I can get into it a little bit.
Yeah.
So usually my understanding is that, and like we've chatted about this in like my training a bit, and I'm not an expert, of course, but one day I hope to be. And the like usually there are three big phases of clinical trials. Like once Sam's done all the like making everything, then and the medication comes to like me, for example, if I'm the doctor running the clinical trial eventually. Like the first, or like maybe with a pharmaceutical company or as part of an academic institution. The first thing is what Sam talked about, like phase 1 trials, where you're doing it in a small group of people to see whether it's safe primarily, because safety is the number one thing. Efficacy comes next. Then like that's phase 1 usually, and sometimes like if you see that it's effective, especially in like a population of like cancer patients, for example, who like need the cancer medication, like oftentimes you're doing it only in people who, like for cancer, for example, you're only doing new cancer treatments for people who have no other option, just because of the ethics of it, because like they like you really want to give people things that have been proven to work as compared to things that haven't been proven. And so like if you see benefit in that, like in a phase 1 study where the patients are really like they have no other option and this is just something new to try, like that's really exciting. However, you can't necessarily expect to see any benefit in such a small number of patients. And so then you move to phase 2, which if the drug is safe, and if you like have a sense of like how it is like broken down and used by the body, which is something that phase 1 trials are often used for too, or the big term is pharmacokinetics. Then you move to phase 2, which is more like looking at a larger number of people to see if it's both safe and effective. And then if it is both safe and effective in your phase 2 trial, then you move to phase 3, which is much like a larger trial, even larger, and you're looking for it to be safe and effective again, and also to detect for more rare side effects that you might not necessarily have seen in your phase 2 trial, where like that had a relatively smaller sample size. And if you send all this data like in that very big packet to the FDA, and they review all that, and then Sam hears back about all this stuff, and if let's say there was a like, oh, maybe the medication caused a fever, we didn't expect that, like let's go, correct me if I'm wrong, Sam, then you go back and inspect stuff. Ideally you don't want that to happen at all, and that would never happen, or I shouldn't say would never happen, but
I should probably clarify a couple things here too.
Yeah.
So now that we've talked about phase 1, so first off, I should say I don't do manufacturing particularly. Manufacturing is done by another group of people who, I understand how things are made and how to make things, like in a theoretical sense, and like the best way to make something. But the actual execution of that is a different skill. And there are other folks who do this. It's also very tedious. There's a lot of folks who do that for a few years and move into like the process development stuff, which is great for me because I get to learn how things are made, because I haven't actually done that myself. But it's very tricky work. It tends to pay very well, but it's also like it's manufacturing. It's working in a factory, in a clean room, in a clean suit for say a 12-hour shift. Not necessarily 12 hours, but it depends on what's going on. It's a long time, and things can go wrong, and you need to fix them, and it depends on how companies are organized and everything else. So, I don't do that. But also, before the clinical trial happens, there's another packet that goes to the FDA called an IND application, initial new drug. And that IND is before the phase 1 data. That is what PD teams spend a lot of time working on, is saying, this is what we did and why. This is like, that's when I say, oh, you see, you get all that test data. That also goes into that IND application. And this also goes by different names. IND is the FDA name for it, but a lot of companies will test overseas, different pharmaceutical, but depending on where you live, it may be called something different. I forget, like, I don't know all the overseas names for them. I've heard a few.
Yeah.
Because companies tend to test internationally for a variety of reasons. Patient population, easy to, it's complicated. But, yeah, so that's the first set of paperwork you send to your regulator. And then you get all that data back and you report on your results, like you were saying. And then you say, then you say there's a fever or whatever. And then you go through the data and you go, was there a problem with the manufacturing? Is this a side effect? Could this be a thing that happened with the patient? So, one thing about trials is everything that happens to the patient is recorded. So if the patient gets a cold while they're on your drug, that still gets reported in the application packet, and then you have to sort those results. I'm not involved in I'm not on a clinical team, but I'm aware of you get kind of I forget the terminology, but it's
go ahead. Sorry, Sam.
It was adverse events is what they're referred to.
Yeah, and you want to see is there a greater frequency of adverse events in the people who receive the drug as compared to people who receive the placebo, for example?
Yeah, not every drug is placebo controlled either, and there's reasons for that. You don't want to.
You can't. There are some situations where you can't give people a fake drug. Like you can't
ethically or it's usually an ethical reason. I was gonna say what was the other thing? No, I was also thinking about something else too. But yeah, a lot of times there's a placebo. You have to, because you want to have a control arm. There's a few cases where you can't do placebo control for a variety of reasons. Also, how you formulate the placebo, what it is. Is it just saline? Is it formulation buffer? Is another question. So the part of this I skipped over because I was talking for such a long time is also when you make the drug, it has to be in a specific formula. Is it in a pill? Does it get injected? How does that happen? What's the chemicals in there? You can't just take a protein and inject it to somebody. You just have just goo. Proteins tend to be solid when you can't just have dust or goo going to somebody. And so that's a whole other thing. And that's you want it to be stable. You want it to be the right pH. You don't want to hurt. People who put citric acid in and that hurts going in because it's acidic. Citric acid, by the way, is it's like lemon juice. Yeah. But we're talking way lower than that. We're not injecting people with lemon juice, but it's that would hurt. But it's used as stabilizers, and there's a whole bunch of other things.
Fascinating.
Yeah, and so sometimes you want to ask. Sometimes a question with a placebo control is, "Hey, is this because of the stabilizers, not the drug?" That's another question you can ask. Do you inject them with the mix of stabilizers you put in there, and then sorry. The thing I was thinking about with the placebo is sometimes you do a placebo-controlled test and then you inject the people with the placebo. You'll go back and give them the drug. Yeah. Which also gives you a great baseline because now you know what their person is like. Person gets migraines before they get the drug, and then they get the drug and they still get migraines. Okay, that's just the person. That's not going to give them. It gets really complicated. But with the adverse event reporting, what I was saying is you sort based on whether or not you think it's based on the drug. They all get reported. And then it's, is this because of the drug, like you were saying? Yeah.
Yeah. I feel like we could eventually, I think, once I personally have a little more experience, I would love to do a whole episode on clinical trial design. That would be cool. Not that we can have it as actual, medical advice, but I think that would be something cool.
It's also, dear listener, if you're not working for a pharmaceutical company, you should not be running a clinical trial. Or if you're working for a pharmaceutical company, an academic institution, you don't have all those people there. You should not be running a clinical trial on other human beings.
Really?
No. Don't try this at home.
Please don't try this at home. Trust me, you don't have enough nerds arguing about this to try this at home in any proper way. I guess, okay, if you live in a giant commune full of pharmaceutical experts and also have several gazillion dollars lying around and a clean room, I guess you can do this at home. If that doesn't describe you, dear listener.
Sam, is that your dream? No.
You can neither confirm nor deny. So you do all this clinical trial stuff. You also start asking questions too. So, I don't know. You've done all of this. Imagine now you have a drug and hurrah, it works. You can make it. It makes sense. The FDA says, yeah, this works. You probably argued with them a bit because they're going to have questions. That's their job.
Yeah.
Great. Now you got to do the part of actually getting it into a person.
Mm-hmm.
Like, so you've done a clinical trial, which is a huge logistical thing.
Yeah.
Right? You have to be able to ship the thing. You have to be able to confirm that it is the thing the whole way. You have to have supply chain management. You have to make sure the doctors are trained how to use this. Sometimes the drug is like, what the heck am I going to do with this? And they're holding up something they've never seen before.
Yeah.
Sometimes you have to argue with the doctors that, yes, you have to use a thing you haven't seen before. Please. This sounds silly. It's happened. Then you now have to do several things. First off, you have to scale up even farther. So now you've had a pharmaceutical, you've got your giant vat of hamster cancer. You know, it's the size of a Toyota Prius. Congratulations, you now need to make a vat of hamster cancer the size of like a grain silo. Okay, that's an exaggeration, but you might just do it multiple times to get that much stuff.
Many Toyota Priuses.
Many Toyota Priuses. Imagine, dear listener, not one Toyota Prius, but many Toyota Priuses. It costs as much as a Lamborghini, and it takes up the same amount of space as a Toyota Prius.
Many Toyota Priuses that cost the amount of a Lamborghini. Is that what you're saying?
Sure. No, more than that. You just picture a lot of money, now double it. You now have this question of, now you've made a biomolecule. Biomolecules, if you could tell from the name, tend to exist in biological systems. You take them out of the biological system, you put them in a thing that you think is relatively stable. Now you have to ship it. And if you remember with the COVID vaccines, which were biotherapeutics, they were mRNA. So they were produced, I believe, using in vitro translation. So they weren't made in cells, but they were made...
it was there. From what I remember, like, I'll have to double check, but it was a lot of... It was not synthesized using organisms.
Yeah.
They had, like, special machines that made it, like, base by base in the DNA, or in the RNA ladder.
Which, by the way, if you're talking about purification and stuff, now your impurities are things that are misspellings, essentially. You still have to get rid of them.
Yeah.
So now you have this thing, and the fun thing with the COVID vaccines was when they first made them, they had to be stored at essentially, like, minus 80 degrees Celsius. So that's, like...
Very cool.
Very cool. I think it's like minus 100 Fahrenheit or something ridiculous.
It's like, if you open a minus 80 freezer... and you grab onto a thing that is minus 80 degrees, you will get a freezer burn if you hold your hand there long enough.
Okay, yeah, if you hold your hand there long enough.
Confirm from personal experience.
I was going to say, because if you don't hold your hand there long enough, it's okay. We don't need to go into why we're both too stubborn to wear gloves in a minus 80 freezer.
I was wearing gloves, I think.
No, the big mittens.
Oh, yeah, no.
Yeah, the big mittens... There's these big... It's a whole thing. It's hard to grab. Imagine, dear listener, that you're now trying to pick up a Toyota Prius the size of a small Lego piece. And you've got roughly the form factor and size of a thing you're now trying to pick up with the thickest mittens you've ever worn. You'll understand why we've both touched a minus 80 freezer with a latex glove on. So, yeah. But those freezers are expensive. They're complicated. If you need to ship something in a freezer like that, when was the last time you saw a truck that was cold as minus 80? It's very special equipment. It was a pain in the butt. This is incredibly challenging logistics. There are entire teams at a pharmaceutical company, regardless of whether or not it needs to be that cold, who are in charge of just determining, how do I ship a thing? I'm not on that team either. I've talked to all of these teams I've talked about, though, I've talked to in the course of doing my job. Process development is very fun being in the middle of everything, and you have to talk to everybody. I love that. You have to work through everybody's concerns. You have to make a thing that works. If you ever buy an IKEA bookshelf, it's not just about making a bookshelf. It's also making a bookshelf that flat packs and will fit in the back of your car. Also not fall over. Also not catch on fire. Also be buildable. Those are all questions. The same thing goes for a drug. So one of the things with the COVID vaccines is they actually just tested what happened if you stored it at warmer temperatures, and it turns out it was okay. But when you make a drug like this the first time you're doing it, especially if you're in a hurry, like you are when there's a global pandemic, you have to start off with, okay, what is the safest thing you can possibly do? The safest thing you can possibly do is store it at the cold temperatures that we used in research. Yeah. You do that. Then you say, what if I store it slightly warmer? What if I store it slightly warmer? What if I store it, what if I store it, instead of storing in a special freezer, I store it in a normal freezer? What if I store it in a fridge? What if I just leave it at room temperature? And there are teams that test this. it's all part of this process, and like that's towards the end of it. And you also try to figure out how to make it cheaper. You try to figure out how to make it more effective, potentially. Although that then turns into a whole other thing, because if you change the formulation, it's a new IND, and typically you make changes between phases of trials, small ones, because you can submit like modifications. Once the drug's on the market, really, really hard to change manufacturing processes. The FDA does not like that, because you might change the product and then you have to prove that you haven't, and that means that data packet gets bigger. And you have to argue with the FDA. And this is for good reason, because this is getting injected into a person, and if you have a thing that you know works and is safe, you don't mess with it. Because, this is all very serious business. But yeah. I think the question then goes kind of like, where from here, right? I don't know, Joe, if you have any thoughts. Or now, you haven't said very.
I think it, like, we're gonna keep making very cool new biologics, and like a lot of the principles that we've learned along the way are going to stay the same. And as we develop new types of biologics, I imagine processes will change.
Yeah.
Like there was a point where we didn't have big vats of hamster cancer. And so, like, where, as Sam alluded to, we were getting like insulin from like animals at one point. So it'll be very curious. I can't really imagine how things will change, but maybe we will start to do more things like in vitro translation, where instead of like making things in like from a cell, you have all the pieces from a cell that actually like make the protein. Yeah. That could be interesting to see adopted more widely.
Dear listener, I have several degrees in biochemistry. I always screw up transcription and translation. I've done it in this episode. I don't know why. Transcribing and translating are words that exist outside of biology. They have specific meanings, and they align with what's happening in a biological system. If I said one of those words in the course of this episode, it was probably used in the wrong context. Joe is better at this than I am. He's a molecular biologist.
We've been primarily talking about translation, where you're making proteins from RNA that has been transcribed.
In vitro transcription is how mRNA vaccines are made. Then, yes, not in vitro translation.
Mm-hmm.
Wow.
Yeah, no worries. They both start with the same thing. So it's A-okay.
We're doing great over here.
I've learned so much on this episode. I'm gonna be real. Like, I think both Natalie and I have learned quite a lot. Like, I think we were both blown away by the fact that, like, I don't know, like, as someone who takes like multiple medications, it's really interesting to know that the way in which, like, it, like, to be fair, am I taking biologics? I am not currently taking biologics.
Do you have to inject yourself with something regularly, or do you go to a site to be injected with
something? I do not, but my grandfather did. And he was one of the, like, he had diabetes, and he would inject insulin every day. And, like, seeing him have to deal with that was one of the things that got me interested in science. And I always wondered, like, how that was made. And it's reassuring to hear that when we're making things, we do like special tests to like experiment and make sure that the things that we are worried about like actually like can be eliminated. Like, that's a really cool piece of knowledge that I honestly didn't have. Like, I just kind of assumed it, but it's cool to know that, like, based on your anecdotal experience, that it actually happens.
Yeah. So, it's worth noting that, like, I've also seen this process from different viewpoints at multiple different organizations at this point. So it's not just XYZ company. It's I've been in other places. There are commonalities. I've worked with different types of drugs. I've talked to people who, have worked. Also, my colleagues did not just start off doing what they were doing all the time. And there is a degree of like, this thing is very expensive too, and that is always a consideration. But there's always, first and foremost, this idea of you're making something that needs to be safe. Yeah. And that is number one. Number two is it should have its best chance of working. I've definitely said, like, people are like, well, what if this doesn't work? I'm like, well, it's not my job to make sure it works in a process team. It's my job to make sure that it has the best chance of working in a patient and that it is safe, as safe as it can be. I mean, when you're injecting something new into somebody, clinical trials have a risk. There's a reason why there's a large informed consent process, and a lot of people are involved in making sure that patients are informed of that, and it's because, everything comes with risk. Drugs especially come with risk. And if something hasn't been put in a person before, like, there have been instances where that risk has been severe. But, that's also part of the process. And by the time a drug gets to somebody who's not sitting there just having the risk discussed with them by a physician, it has gone through people like that. And it is also, like, you have to say something about people who sign up for that, especially for when there's studies that test safety first and it's in a healthy patient population, which happens. Those are folks who are perhaps braver than I am. And they go into this knowing that there is a risk, and they do it anyway.
I knew a number of people who volunteered for the mRNA vaccine trials, for example.
Yeah.
Back in the day. Wow, it's been a few years, hasn't it?
Yeah, but like, that is a risk, and folks were willing to take on that risk knowing that.
Yeah.
There's a lot. There are so many people involved in a medication. I've worked on a medication, left the company, been at a house party, ran into somebody who was working on the commercialization team. I had never seen this person before. I haven't seen him again either, to be honest. Like, there's so many people involved in making a single thing work who have a lot of specific skill sets.
Yeah.
And that's good. Some of them are very headstrong about it, and that's also good. You want somebody who's willing to fight everybody else about a safety issue. It happens. Sometimes it's their opinion of how you test a thing. It's like, no, we're going to test this thing this way, and here's where we do it. And it's like, well, what if we can't? How do we work through this? I've had arguments with people about the measuring error on some of the devices we were using. That, like, I eventually ended up going to the lab and testing it, because I was like, okay, how do we assess this?
Yeah.
And we're talking, like, fractions of a milliliter for, like, a large volume, because people care. Yeah. Sorry, just to say all that. Like, there's so many people involved in this. And, like, the future of this, I mean, the industry right now is in a weird spot, I will say. I think I made a joke about that at the beginning. Obviously, there are still folks working on these things. I'll probably be working on them again in the near future. There's a lot of money involved, and that's because it's meant for a damn good reason. Pardon my French. Because you need to be able to make sure that you are making something the right way. And doing that well. It doesn't always mean it's perfect either. Like, but it's certainly I've never ever been in a room where it's not the best everyone there could possibly do.
Yeah.
And you're talking like a century worth of education and quadruple that of experience just of how many people are in the room. Like, yeah, like there's more nerd power in those rooms than might have existed for, like, I don't know. Like, I'm sure you've used an app that's been developed by a tech company that didn't even have as many people as are on a PD team in the room. I'm not sure Uber had as many nerds looking at something by the time it launched than did in a pharmaceutical company before a drug even got to clinical trials. Yeah. They're huge organizations. There's lots of people, and it's a really cool process to see. It doesn't always work out. Some of the drugs I've worked on are on the market. Some of the drugs I've worked on are never getting there.
That's how it goes.
Well, that's a good thing, because if they don't get there, that means the system
is working.
And if they do get there, it means the system is working. It's the system. It sucks because sometimes, it's 10 years of research and $500 million and it didn't work. That's life.
Better to be safe than sorry.
But also, now.
Yeah.
There's a whole team of people now that know, hey, this approach doesn't work.
Yeah.
Next time someone brings it up, in a room, next company they're at, they're gonna be like, hey, that doesn't work. Like, and that's good. That's how these things go. And keep learning, keep moving, and if you look at like cancer survival outcomes, it works.
Yeah.
I've got an uncle who would not be alive if he had got cancer 10 years earlier. Because we figured out how to fight it.
Yeah.
And we figured out how to make the thing to fight it. And it was not a simple drug. It was a biotherapeutic that was very complicated. More complicated than the stuff I described. This was the simple version. We're like an hour and a half in, and it's the simple version.
I apologize if this is insensitive, but did they use hamster cancer to fight the cancer?
I actually don't remember the precise drug he was on. There's a couple of things that were in my head, and I can't particularly remember. I do know like the classes of things.
I'm sure that's happened 100%, at some point, Joe.
Yeah. Oh yeah, yeah.
And I say hamster cancer in a vat. I mean, those are cancers. Those are cells. Cancer cells multiply.
Yeah.
The other thing is, I probably should have said this at the top when I made a joke about hamster cancer. They are not capable of multiplying human beings. They're hamster cells. And secondly, they don't even get to the first stage of downstream. They got caught in that filter I was talking about that made it into orange. There's not a cell of that nasty smelling orange Gatorade. There's not a single hamster cell in there. I've never been handed a hamster can. I've worked with this stuff before, and I've never been handed a single. I would bet that there probably wasn't a single one of those cells in there, because that's the point.
Yeah, and also the point you brought up earlier, like even if you get like one or two in there, there are so many downstream processes, and also even if it was in the final product, it's not really going to do much.
Yeah, and also
from my understanding.
Yeah, I mean, if there was 1 million of them in there, there's probably a 2 million fold reduction. There's a 50% chance one cell would get through.
Yeah,
in the entire batch potentially. Right? If we're talking, if you have 1 million of these contaminant in the start of the batch, and you have a 2 million fold reduction. If you've reduced 2 million times, it means there's a coin toss that a single cell got through.
Yeah.
Right? The whole process, you control everything you conceivably can, and then you test to make sure you didn't mess that up.
Yeah.
Yeah.
Well, I think I for one am really grateful for you sharing your experience.
Yeah, thank you so much for taking the time.
Yeah, because I know you've been wanting to share with us and our listeners for a long time, and it's helpful for me to hear as someone who kind of like I do a lot of the stuff. Like, I will probably do some stuff, like, before on the research side and a little bit after on the actual, like, clinical trial side. But seeing what happens in between is really cool and really important. And I think what a lot of people don't really know exactly how their medicine gets made. And so it's really, things like this, I think, are really important for, I mean, I think a lot of people assume that people who are even in medicine know how the medicine is made. And that's not always true. And so it's really helpful for me to see, too. So thank you.
Yeah. And every one of them is different, too. So I can't speak to everything. Yeah.
But the general principles.
And the other thing is I can speak to the folks who work in that, too, in the ethos, like the vibe. And I've worked in companies that aren't manufacturing therapeutics. They're manufacturing other stuff in the biospace. And it tends to be more lax. Not, like, sloppy, but, like, if it's not getting injected into a human being, you're not going to be as concerned about it. And I actually really love that level of precision. I love, having that drive to make the best thing possible. That's very satisfying. I'm a nerd about that, and I like being surrounded by other people who are too. And yeah, I think that's the other thing is like, especially right now, people are very anxious about medication and these other things, and like, yeah, there's a lot of other things that happen in pharmaceutical companies that aren't making medications, and I can't necessarily speak to those things. But as far as the actually making something side goes, I know the folks who work on that, and I've worked with people who I wouldn't say I'm friends with them even, but their willingness to go the extra mile to make sure the thing is the best possible is really apparent. And that's like seriously. It's taken seriously, and these are sometimes like it's really funny because like sometimes these folks outside of work, you would never know that they're like that. But the second it becomes a professional environment, like there is these serious discussions about what is happening biologically, what is going on, and that is how you gab a job like that. And it's very cool.
Oh yeah, absolutely.
All right, listeners, we've been recording this for a while. It is late. It is late o'clock Eastern time. Natalie, you still have sunlight because you live in the earlier part of the country.
I live thousands of miles away.
I'm sure you're going to go have like dinner now or something, and I'm going to go to bed.
Yep.
But thank you for joining us. I hope you learned some things, and Nata, if you want to talk about where you can find us online, and
we'll see if I can. At Interactome Media for Twitter, and that's not true.
I believe it's...
Let me... At the Interactome on Twitter, and then Interactome_media on Instagram.
We have
that
a
is
Mastodon
true.
account on, I think, Universidon or something. I'm in charge of that. If you have a question about this episode and you put it on there, I will reply. I'll be shocked to get a notification from that account, but I'll reply to it. If you guys even know what Mastodon is, I don't know. I'm a weird nerd. I should go to Blue Sky and get on there. Also, if you post anywhere else, any questions, if I can answer them, I would love to. And I'm sure Joe and Natalie's the same.
Catch you next time.
See you guys.